Okay, my name is Eduardo Mongalo. I'm presenting on nanoscale red blood cell substitutes. So what are red blood cell substitutes? They're basically artificial red blood cell red blood cells, and pretty much how they're engineered and made is that think of it as what they do is that they get a small molecule, and then with that molecule they're able to produce even more and more, many of them. So think of it as now what they make is polyhemoglobin. So they get hemoglobin, and then what they do is that they produce even more of it, and what they do is that they could even produce a protein membrane, polymembrane. So that's basically what they do, is that they try to multiply huge amounts of it. So the structure of a hemoglobin, on the top right, you can see that on the chemical over there, you can see that in the middle, you can see that an iron ion. And with hemoglobin, the purpose of it is that it carries oxygen, and you can see 4 chains. There's 4 heme groups, the chains, and every hemoglobin carries 4 oxygen, since every chain has an iron atom. So that's the function of it, is that the blood cell, the hemoglobin, is that it carries the blood as you breathe in the oxygen. The hemoglobin carries the blood all throughout your system and then drops it off for nutrients and so on. So then now on the bottom right, the hemoglobin. So the hemoglobin, the polyhemoglobin, you can see on the bottom right is that they're connected to each other. So think of it as like packages. So within the membrane, the protein membrane, inside are the packages, the polyhemoglobin. And as you can see is that you can see CAT catalase, which is an enzyme that it's good for protecting from oxidative damage to the cells, to the DNA, to protein. And what they do is that they add in some enzymes to protect, the body as it goes through the system. So here's an example, just a small example of the membrane is the polypeptide chains. And as you can see, just the formula, you can see that how they're made is that they connect to each other. The hydrogen and the hydrogen monoxide on the right, on the left, you can see that they disappear once they chain together, and it turns into water. And so then how they're made, the membranes, is that they, the same thing, they multiply it. So they repeat the sequences of DNA, cutting them at different lengths. So perfluorocarbons, it's another example. Instead of hemoglobin, it's with perfluorocarbons, and they're really good at transporting oxygen throughout the body. And since they're pretty small, well, both of them are small. This one is pretty good at fixing tissues, smaller tissues around the area. I'll talk about them. So what can RBC substitute do to help in medicine? So replace donor RBCs. There's not a lot of RBCs everywhere, especially in smaller countries, poorer countries. I mean, they don't have enough for supply. So what you could do is that you get the supply for the red blood cells, and you can produce even more for the substitutes. So they could treat infective agents, stuff like bacteria and viruses. So an example is HIV. During that time in the 1980s, people they were being contaminated. As they were given blood, they were being contaminated with HIV, and with red blood cell substitutes, they could prevent that, and they could take action in hemorrhagic shock, which is a lot of blood loss in the system patients get. And although it's still a serious matter, but it's more of like the supply that they could get, the doctors, to help the patient right away. And then other emergency transfusions going on, they could help treat traumatic brain injury. So the swollen tissues, since the perfluorocarbons, they're small enough, they could treat the swollen tissues. some downside of RBC substitutes. So they can't stay inside for long periods of times, and also they disappear once the patient is injected with something. And but the good thing is that they could be stored for up to a year, and normal blood cells, they can't be stored. They are stored up to 42 days at lower temperatures. And so a bad thing about RBC substitutes is that they can bond to nitric oxide. Nitric oxide is responsible for the pressure in your blood, and since the RBC substitutes are small enough to enter and react with nitric oxide, they could mess up the blood pressure of the patients, and high levels of blood pressure, and that could result into death. So those are bad sides, and here are the sources. So just wanted to talk about why I'm interested in this topic. It's because I'm interested in this topic because although I'm studying mechanical engineering, I'm thinking of studying mastering in biomedical engineering. And although I think it's still not in the study of this, I'm going to do more in designing of medical equipment. But I think just this whole thing is really interesting, how engineering could impact and some fields, they could impact a lot of lives. And I think it's really good for the future, and that's what I really hope for, is a bright future for advanced technology to help people around the world in poor countries resolve into peace. So that's it.